Lovely. Okay, this one. All right, can you see that? Good.
Yep.
Right. Oh, not that. Okay. Welcome, everybody. This is week 2 review session. We're going to be covering the first half of the material for this week. So before we get started, does anyone have anything specific that they want to go over or put more focus on?
I just have a question regarding the problem set, just the wording.
Yep.
When it's asking for chemical categorizations, like what does that really mean?
Which question?
It's on both A and B for question 1. When it's like saying give specific examples as well as chemical categories. Oh my god, I can't even say it, but like, you know what I mean?
Hold on, I've just pulled it up.
Yeah, me too.
Right. So chemical categorization. Oh my god, categorizations. I think what she means is like polar, non-polar, hydrophobic, hydrophilic, charged, uncharged, that type of thing.
Yeah, exactly. Cool.
Does that make sense?
Yeah, thank you.
Okay, cool.
I also had a question on the problem set. More like the third question, they asked like what makes enzymes different from regular proteins. So I don't know if that's what we're going over in this review session, but that would be helpful.
I think Michelle wanted to cover that Friday, but we can go over the third question on the problem set at the end if you want.
Awesome. Yeah, that'd be great. Thank you.
Well, we'll go over that a little bit, right? KD and stuff.
Oh yeah, true. We are going to go over KD. So
yeah.
Okay.
But we can also go over that question too at the end.
Yeah, definitely.
Awesome. Anybody else have any quick questions? Otherwise, we can get started. Cool. All right. Welcome to week 2 review session. I'm Jason. That's the other person is Rachel.
I'm Rachel. Hi.
And yeah, today we're going to be discussing the first week stuff, protein folding, KD, stuff like that, how to fix misfolded proteins. And yeah, Rachel will start it off.
Okay. Wait, Jason, can you hear me? Any chance? Yep. Okay, perfect, because my computer has been glitching, but okay. So first thing we're going to talk about is protein folding in the environment. So basically, this is just a whole thing on Professor Francis's structure equals function. You're going to see this a lot throughout this course, specifically here. So the environment affects a protein and can significantly impact its function. So this is why cells control environmental factors such as temperature, pH, salt concentration, because these factors can cause denaturation, which is just a fancy word for an unfolded protein. So basically, there are proteins in the cell called chaperones. We kind of talked about that today in class that help proteins fold or correct them when they are unfolding. So we think of these like chaperones on a field trip. They help kids behave. These are the same, kind of doing the same thing. They're helping proteins behave and fold improperly, or help them fold properly, or help them fold when they're being they're not folding properly. So just a side note, while we get into this, for this class, we consider covalent bonds to be the strongest bonds between molecules. So an example would be a peptide bond is a covalent bond. In actual chemistry or biochemistry, this isn't always the case, but for this class, it will be the case. So this will be the strongest bonds between molecules. So most of the time, if you're asked about covalent bonds in the environment, you're going to assume that covalent bonds are too strong to break. So what this means is if there's like a really high temperature, if she gives you a scenario, and like there are covalent bonds, it wouldn't affect the covalent bonds for this class. So that's just something to keep in mind while we go through this. So thermal energy or temperature affects the stability of non-covalent bonds. So like I said before, with every bond that I talk about here, it's going to be just non-covalent because we will not be affecting our covalent bonds because they're too strong to break. So temperature can affect the stability. Like I mentioned kind of before, they can mention they can affect the stability of covalent bonds too, but only in extreme conditions. So not under normal conditions in our bodies. So in normal conditions in our bodies, the temperature is not high enough to break our covalent bonds. So just a little side note here, less thermal energy means more stable bonds. So the more energy you have, the more unstable your bonds are going to be. But we also talked about this in class, how there's this Goldilocks hypothesis where being the most stable is not always ideal. So sometimes it sounds like being the most stable is something that you would want to obtain, but depending on the needs of the cell, this isn't always the case. So for example, if there's too little thermal energy, the diffusion and motion may not be enough for interactions to occur between the molecules. And if there's too much, you may affect the stability of too many non-covalent bonds. There may be too much breaking, too much protein unfolding. An example is kind of when we have a fever and our temperatures rise. This can cause proteins to unfold in our cells. That's part of the reason why we feel crappy when we get a fever. So this Goldilocks hypothesis kind of just emphasizes that our bodies have evolved to find conditions that are just right for our proteins and cells to function properly and without too many like problems such as like unfolding, denaturing, or too little thermal energy and all that stuff. So that's just what that means, that Goldilocks hypothesis.
Cool. I hope you guys like my meme. If you ever need a pickup line, this is a great one. It worked for me. So
no, it did not.
It did.
Emily did it. Is she here?
All right.
Hi, Rachel.
Hi, Emily. That is so funny. Oh my God. It is Valentine's week. All right.
I have a quick question about the last slide.
Sure.
I'm just a little confused because I know you said that when asked about covalent bonds in the environment, then assume that they can't be broken, but I'm just confused when it says like can affect the stability of covalent bonds, but like only in extreme conditions. So like if she gives us a problem where she states that it's an extreme condition with the temperature, like then would we not assume that they can't be broken?
So for all intents and purposes of this class, covalent bonds are too strong to break, no matter the condition. If you do get an example like that on a test, I would clarify with her. So like during your test, I know you guys haven't had them yet, but basically Professor Francis will be accessible by email throughout the entire time you're taking your test. So you can just shoot her an email and she will clarify that for you if she does give you a specific question like that. But just remember that stability does not always equal breaking. So the extreme conditions could affect the stability of the covalent bonds, but they may not break. When we talk about the strength of covalent bonds in this condition, in like these conditions, we're just saying that in these environmental conditions, anything that we talk about, the covalent bonds are too strong to break. Does that kind of make sense?
Yeah. Okay. Thank you.
Yeah, no problem. But definitely clarify with her if it ever comes up on a test and you're like, is this one of those conditions where it does break or something? Because she'll be sure to clarify that for you. Okay. So another two environmental conditions are pH and salts. So the pH and salt concentration in a cell can affect the ionic or the charged interactions between molecules. So I know you guys, this is still the second week of class, so you don't know all of the amino acids yet, and you won't be expected to know all of them because you'll have like your charts available to you if you need to look at it during an exam or the problem set. But basically what this means is that some amino acids that are more acidic will be negatively charged at a specific pH. And then there's some amino acids that are more basic will be positively charged at a specific pH. So that's just what it means is this pH in the cell affects the charge on certain molecules such as amino acids. And then another example of something that could be affected are the disulfide or the salt bridges in tertiary structure. So these are just things that these environmental factors affect. Another thing that we don't really talk about that much, but can be brought up on a test or something, light energy can introduce another form of energy to the system. This only affects non-covalent bonds, and higher energy wavelengths can disrupt these non-covalent bonds. So these are like UV wavelengths will probably disrupt non-covalent bonds. But these are just environmental factors that affect our bonds and our molecules in our cells. Does anyone have any questions before I move on? No, cool. Okay, so protein unfolding. We talked about this a little bit, but what happens when proteins unfold, and this is what makes it problematic, is they're hydrophobic R groups that were tucked away on the inside, which is also known as the hydrophobic effect. These are now exposed to the polar aqueous solution that surrounds the proteins. So we don't want this because they are hydrophobic and they don't like aqueous solutions. So what happens is when proteins unfold, and now that this hydrophobic core is now exposed, they will bind to other hydrophobic R groups on other proteins as kind of like a way to like bind to something that's more hydrophobic, because they're trying to avoid having to bind or be exposed to a polar solution. So instead they will try and search kind of frantically for other hydrophobic R groups on other proteins. And this will lead to protein aggregation. So a way to think of this is when you cook an egg, you know how it turns kind of from that gooey liquidy to a solid. That's protein aggregation. You're exposing hydrophobic cores of the proteins in the egg to the aqueous solution surrounding it, and they will just aggregate as a way to cope with that. There's also evidence that protein aggregation can lead to neurodegenerative disorders such as Alzheimer's due to the accumulation of tau or alpha amyloid proteins, which are the proteins involved in Alzheimer's. But yeah, so this is just what happens when you get proteins to unfold. They start to aggregate, and it's not always a good thing. It's usually a bad thing. But the cells have certain responses called stress responses to kind of fix this problem. There's two. There's the heat shock response, which occurs in the cytoplasm and the nucleus. And then there's the unfolded protein response, which occurs in the ER. It's important that you know the two take place in different areas in the cell, because that'll be important when we get to those slides.
Cool. Any questions about those? Otherwise, we can move on to how to fix an unfolded protein. Cool. All right, so what happens now when the protein doesn't fold right, or something, it gets messed up by one of the things that Rachel was talking about, like temperature, pH, or whatever? So some key points to consider. So cells have, like, it would be really bad if lots of proteins were unfolding or misfolding all the time. So cells have ways to fix this, or just destroy the protein if it's not working. So if it's sent to the proteasome, it's probably called the heat. It's probably as a result of the heat shock response. If it's going through the endomembrane system, there's an ER specific stress response we call the unfolded protein response, and I'll go over that in a second. And there's also general homeostasis mechanisms that the cell has to keep things at the general temperature, because like we were saying earlier, cells have a specific temperature that they like to be at. So we want to stay around that temperature. Like Rachel was mentioning, there's a lot of neurodegenerative diseases that are caused by misfolding proteins. One example is Alzheimer's, like she was saying. Another is mad cow disease as a result of prions. Laura doesn't talk about that anymore, so you don't need to worry about that. But just know that it's something that happens, and that's a result of misfolded proteins that form like these clumps. Another thing that Laura talked about in class, when proteins aggregate, they form clumps, and these clumps are insoluble because the protein unravels, and then the hydrophobic parts of that are usually hidden on like the core of a protein, they become exposed, and that changes the solubility of the protein. In order to help proteins fold, there are things called chaperones. So chaperones are, I'm pretty sure they're proteins. They're like these special proteins that help other proteins fold, or to stabilize them when they're unfolding or misfolding. You could kind of think of them as like a chaperone on like an elementary school field trip. They're kind of going along to supervise the other proteins and make sure they don't get out of control. And chaperones also have a job of sensing when proteins are misfolding or unfolding. So we'll talk about in a second. You'd want more chaperones when something is going wrong in the cell. One of the unfolded protein responses is the heat shock response. And so basically if the cell experiences like a rapid change in temperature, it'll detect that something's wrong with the protein, and because temperature affects the protein, so it'll detect something's wrong, and it'll ship them off to the proteasome to get destroyed. The proteasome is just where proteins are destroyed in the cell. In the heat shock response, chaperone production is also upregulated, and this part of the unfolded protein response occurs in the cytosol in the nucleus. This would not occur in like the endomembrane system. Any questions about this stuff so far?
So for the unfolded protein response, would that also have an upregulation of chaperone proteins?
Yep, that would also have an upregulation of chaperone proteins. No problem. That would also have an upregulation of chaperone proteins. I'm going to talk about that in a second.
Okay.
Any other questions? Cool. So for proteins that are in the endomembrane system, so in like the ER, Golgi kind of area, there's an ER specific stress response called the unfolded protein response. Lots of proteins have to go through the ER, so it's important that the ER can detect when something is unfolded. And so basically the main idea is you want to reduce the amount of proteins being created, right? If lots of things are being messed up, you don't want more proteins to continue getting messed up. That would be bad. You want to make more chaperones to be able to help the misfolding and send things where they need to go. You want to make space for more proteins. So you can, there's a couple ways that the cell does this. Not necessarily in a specific order, but just generally things that the cell does. So one thing would be to decrease general translation of proteins. That way the ER has to deal with less folding of proteins in general. Another thing would be to, like we were saying, upregulate chaperone production. So if the proteins are unfolded or misfolded, you would want to upregulate chaperones to help that process. Another thing that may not be super intuitive is to increase lipid biosynthesis. That just means to increase, it basically just increases the size of the membrane. So the membrane is made of lipids. So in biosynthesis means just to make something biological. So you basically just make more space, make the membrane bigger to increase the space in the ER so it can hold more protein. And also, if there's too many proteins in the ER, you could just move the proteins out of the ER to the proteasome by a process called retrotranslocation. Retro just means backwards, so move it back out of the ER to the proteasome. Any questions about the unfolded protein response? Okay, yeah, this is pretty important process in the cell. I don't know how much Laura asks about it, but it's a very important process in general. So some main ideas that we've talked about so far, there are a variety of environmental factors that can affect the protein's folding and function. A big theme in this class is that structure relates to function. So if a protein is misfolded, it has an altered structure, and that probably affects its function. So you want to make sure that the protein is shaped how it should be. Some environmental factors that could affect that, like we talked about, are temperature, pH, salt concentrations, light energy, anything that can mess up the fold of a protein. The biggest thing is that these environmental factors don't affect covalent bonds, at least that we'll talk about in this class. So just think about like what kind of effects that would have on the protein if it's not affecting the covalent bonds. and also like Rachel was talking about, Laura's Goldilocks hypothesis, it's not always best that a protein has stable bonds, because sometimes you want to be able to break a bond, or a bond is only temporary, so you wouldn't want it to always have a covalent bond. That's why the cell doesn't always make things covalently bonded, because to protect against these environmental factors, because you don't always want to have a super strong bond. The term for when a protein unfolds is denaturing, and when it refolds, it's called renaturing. And yeah, when it's really bad when proteins are unfolding or misfolding. So we want to be able to have a response to fix this, and that's the unfolded protein response, or the heat shock response, depending on where it's located in the cell. Any questions? Otherwise, we're going to talk about KD next. Cool.
Okay, so protein ligand interactions and affinities. So I think we started talking a little bit about this in class today, but the thing with enzymes is Laura never really goes into them like too much detail. If you ever take like a biochemistry class like Biochem 420, you'll learn a lot more about them. But we'll just go over like the basics here. So a ligand is just anything that a protein will bind to. Interactions don't always involve a binding pocket. It can be a flat interactive surface, which we don't have a picture of here. But in either case, a protein needs to be oriented correctly, as well as the amino acids exposed from the pocket. Otherwise, you won't get like a good binding, and you won't get the interaction that you need to occur if it's not oriented correctly. The affinity is just a word for the strength of the interaction between the protein and the molecule it's binding to, or the ligand. And the affinity is dependent on the number of bonds between protein and ligand and the strength of those bonds. We're going to talk a little bit about KD in the next slide, which goes over like the relationship between KD and affinity. But that's what affinity is dependent on, how many bonds there are and the strength of them. So KD is the dissociation constant. That's the equation for it. It's just the protein concentration times the ligand concentration over the complex concentration, which is the ligand and protein bound together. But you'll never be asked to use this equation in class. So don't worry about it. The main takeaway here is that KD and affinity have opposite relationships or inverse relationships. So the smaller the KD number, the bigger or the stronger the affinity. So the harder it is to break a bond between a protein and its ligand. So the smaller the KD, the tighter the protein and the ligand binding interaction is, which means the affinity is higher, which means it's harder to break apart the protein and the ligand. The higher the KD number, the bigger the KD number, then you're going to have a smaller affinity, a weaker bond, and easier to break the bond between the protein and the ligand. So KD is a way to measure and describe protein ligand interactions. It's an average rate measurement of how often the protein dissociates from its ligand. So that's just the formal definition of it. But basically what it is it's the concentration of ligand when half the binding sites of the protein are filled. So when the concentration of protein equals the concentration of the complex. So like I said, the smaller the KD, the tighter the bond, the higher the affinity. This also means that it takes less to make more complex. But also, like I said, there's an inverse relationship between affinity and KD. And yeah, the lower the KD, the higher the affinity. That's the main takeaway from KD. Enzymes, I think Michelle talks more about on Friday, but this is just kind of like the intro to enzymes because enzymes are proteins. So when we talk about protein ligand, it's like the same thing as an enzyme and a substrate. That's the last slide we have. It's a pretty short review considering we haven't gone over that much material yet, but does anyone have any questions?
I have a question about sorry, most of my questions are kind of on the problem set.
No, that's okay.
But
I can pull that up 1 second.
Yeah. I mean, before we get into the problem set, could I ask a quick question about the material? Sorry.
Yes.
Okay. So when they say like it goes to equilibrium, what does that really mean for the KD? Does that mean like that the ligand and the I guess I'm having trouble considering like what that means.
So I don't know if equilibrium applies to KD.
Wait, are you talking about the problem today in lecture?
Yeah, well, it was mentioned like when it's at equilibrium, like this KD can be found. Yeah, but yeah, I don't know if that
I think by that she means that the protein, like half the concentration, or the concentration of ligand, or wait 1 second, sorry. It's when the concentration of protein is equal to the concentration of like the ligand in protein complex, I think.
Okay, that seems easier than I guess I was expecting.
I think that's because that's like when you
let me try to find the wording that mixed me up.
Yep.
Okay. So yeah, I think it said mixing ligand and protein will cause them to interact and go to equilibrium. And so that is just meaning that's when it reaches the KD.
Mm-hmm. Yes,
okay.
I think so. Like that's when you calculate KD, is when those things are equal.
Okay. And so I guess when it's at equilibrium, like, I don't know, it just seems weird to me. I don't know.
The biggest thing that she'll ask about it, or like that we use KD for, is the inverse relationship that Rachel was talking about. So when affinity is increased, KD is decreased. So it basically is just like a measurement of how strong the like something is connected to its target, right? And you don't always want a lower KD, because then, like for example, with an enzyme, you don't always want a lower KD because then it can't like release and go help something else, right?
Yeah, I guess maybe my question is like, why does it just stop when it's at 50-50? Because there's still the ligand present.
Like the half thing. Yeah, I don't know.
Okay, it's just like a snapshot.
Yeah,
I think so. Yeah, that's like the best way I could think of it. Even when I took like Biochem 420, we really didn't go into that, so I don't have a better answer for you. But
yeah, I'm just.
She won't ask you to use the equation at all.
Yeah, she'll never ask you to use that.
All right. I guess this is now tying into the problems that where my confusion is, so we can.
Okay.
So, yeah.
Let me share that real quick. Hopefully I'm not sharing the answer key. Okay. So which question first? Number 3, or did someone still have questions on number 1?
Honestly, I kind of have questions on all of them, but the one specifically that I had was 3.
Okay.
Just like when it's asking like the second part, like based on what you know about enzymes, what they do, why do you suppose dissociation constants in the example experiment is used to determine the dissociation? Like, I guess I'm not really understanding what it's asking, because it's talking about KD, and I thought that you can make an assumption about the affinity based on which like direction the KD is going.
Right, but enzymes are special. So what happens when an enzyme does something? Like, is it the same as when it started, I guess?
Yeah, I guess it releases a product that is different from the original substrate. So then you can't like continuously bind and unbind.
Yeah, exactly. So it's something different once it changes into a product. So you can't really measure KD for that.
Okay, so when you normally have you normally have a protein and a ligand, does that like it's
KD.
like a continual process? Oh yeah, so that's the KD. Okay.
Yeah, I just wanted to clarify that last slide. I know I said it was like an intro to enzymes, but what I meant was like the whole like interaction between the protein ligand. I forgot to mention that KD does not work to describe enzyme substrate affinity. It only works for when you just have like just a regular protein and a ligand. So I just want to clarify that.
Okay. What was that? Sorry.
I just wanted to clarify from that last slide that like KD can only be used to describe just a regular protein and a ligand. It doesn't work for enzyme substrate affinity. So when I said it was like an enzyme intro, it's more so that enzyme and substrates do have specific affinities, but they're not measured by KD.
Okay, thank you.
Mm-hmm.
I guess my question would be like, why exactly can we not use KD to measure the affinity of proteins or like enzymes to substrates?
Because the enzyme changes the substrate into a product. So it isn't straight up, like it isn't just a straight up binding / unbinding type interaction. Like the substrate doesn't bind and then unbind. It's the substrate binds in a certain way and then it unbinds as its product. So it's difficult technically to determine the KD for an enzyme substrate pairing because the substrate isn't leaving as the same molecule as it came in as, if that makes sense.
Okay, yeah, that makes more sense.
Okay. You guys have any other questions on the problem set or anything?
Yeah, yeah, all these are a little confusing. Let me try to find.
Can we just like, I'm definitely more of a person that like, I guess I don't like interpret the information like what it's asking me correctly. Can we just like go through like what it's asking?
Yeah.
I like mix up the wording and things and like I don't know.
Yeah, it's definitely a process to get used to how she words things, but so let's see. Rachel, do you want to take this one?
Yeah, sure. So so the first thing I always look at is like the diagram she usually gives you because it kind of helps you look at what she's looking for. So for one, let's see, alpha helices are very common structures, R group, blah, blah, blah. Yeah, discuss what categories of amino acids you expect to find in a protein that interacts with DNA in this way. Be sure to give specific examples of amino acids as well as chemical categorizations of amino acids, discuss the types of bonds. So like I mentioned before, the chemical categorization is just like charged, uncharged, polar, non-polar, hydrophobic, hydrophilic, something like that. So with this specific example, when you see that she mentions alpha helices. So every time you see alpha helices or beta sheets, know that she's talking about secondary structure. So what is always synonymous with secondary structure? Hydrogen bonds. So for this question, you're gonna want to think about the bonds that will be made that interacts with DNA in this like diagram, and this diagram will help you to look at it also. I think one tip I can give you is to remember that DNA is negatively charged. So you want to think about the chemistry involved there. And then you want to deduce what type of amino acids you would see in this type of interaction. So you would want to use this information to kind of determine whether the amino acids are polar uncharged or polar charged or non-polar to answer this question. That's really what she's looking for here.
But also they'd have to be hydrogen bonds because right?
Well, you'd want to mention that what type of bonds that the type of amino acids would make. So yeah, hydrogen bonds would be one of them, but there are some others that they would make as well.
Okay, and just like within the diagram, like it's showing with like the adenine and like thymine. Okay. So I guess I'm just like, where would we attach? Like I'm definitely like a visual learner.
Yeah.
So, like, attaching, like, would it be like on the like left side of the molecule?
I don't actually know what side. I think it would be just the backbone of the DNA. So the backbone with meaning the phosphate like backbone group, or that like 5' end to the 3' end. She doesn't actually like expect you to like know where, but that's where I would assume. So yeah, and then go from there, from what type of bonds it would make with that, and then what type of amino acids it would make with that, and then give like examples of the type of amino acids that like you find would work in this scenario.
Okay.
So say it was like a non-polar. I'll just give you a hint. The answer is not a non-polar. So say you said like methionine, which is a non-polar. Like if you, so say you said the answer was non-polar, then give an example of a non-polar amino acid like methionine. Does that make sense?
Is it asking for like one amino acid that can accomplish both interaction with the negatively charged backbone and a like hydrogen bond, for example, in like the polar base? Or is it asking like, oh, a polar uncharged molecule can make a bond with the middle, or a polar charged molecule can make it one with the outside? I'm just wondering.
More like the second part, like a polar charge, polar uncharged, unpolar charge, whatever.
Okay, so it doesn't have to be like one amino acid to encompass all the categories of.
Okay. No, yeah.
Okay. Thank you.
Yeah, no problem.
Cool.
So for B, the phospholipid bilayer hydrophilic. Let me read the question first. So describe which amino acids you might find in the alpha helix embedded in the cell membrane. Give specific examples, as well as chemical categorization, and discuss the type of bonds that the protein would make. Okay. So yeah, that's kind of similar thing. So you would want to mention what type of interactions would be made, what polar, non-polar R groups, charged or uncharged, basically what interactions they would be making with the bilayer.
Think about, is the lipid bilayer polar or non-polar, and then go from there.
Mm-hmm.
I have another question about this. So are they talking about an amino acid like embedded in the middle of the bilayer, or is it like if you have one embedded on the tip of it? Like, 'cause I know sometimes you can have like proteins sticking out on top of like the cell membrane that are normally like polar and very hydrophilic, but then like you'd also have amino acids, I guess, inside, because it says embedded in the.
Yeah, so it says it says which amino acids you would find in the alpha helix. So she gives you that diagram of what that alpha helix is.
But this last sentence is pretty key. Make sure to take into account the entire chemistry of the phospholipid bilayer. So like it might make different interactions with different parts.
Yeah, depending on where the amino acids you're talking about are.
Okay, thank you. Mm-hmm. Yeah.
And then, do you guys have any more questions on any of question 1 before we go to? No, okay. So question 2, let's see. So it's basically, you're basically talking about what a buffer does, and comparing the changes in KD, and then what would cause this change in KD. I think is what that one's asking for, because she gives you 2 values of KD with 2 different buffers, and then she's just asking you to talk about, remember how we talked about like the environmental factors that affect proteins at the beginning of the review session. She's just asking you to talk about those and explain why or what would cause the KD value to change.
And remember the relationship between KD and affinity. So if KD is lower, affinity is higher. KD is higher, then affinity is lower. So buffer 1 would have a higher affinity than buffer 2. So what can you deduce from that? I guess is kind of the question.
Yeah, I was wondering for the salts, is it basically like the ions are just like getting in the way, like steric hindrance, or is it beyond that? I think in one of her lectures she like had a diagram of like a Cl ion getting in between a protein and its ligand. So is it more of steric hindrance or more beyond that?
And Jason, you can obviously step in too, because I'm not entirely sure, but I don't think salts would cause steric hindrance because they're pretty small molecules. Okay. It would more be like interference, I would say. Okay. So steric hindrance is more when you have like a really bulky molecule.
Yeah. Mm-hmm.
Yeah. And so yeah, that's what I think salt would maybe be more, it would be affecting like the charges or the charged interactions or something like that.
Ionic bonds.
Yeah.
Okay. Thank you.
Yeah, no problem. And then we kind of went over 3. But do you guys have any specific questions?
I think I'm good. I just have to think more about them, but
yeah. Laura has help sessions that are great. I think on Thursdays, or she did last semester. I'm not sure about this semester. Yeah, she does Thursdays and Fridays. Yeah. So she would also be happy to go over anything further that you have questions on, and like exactly what she wants.
Yeah.
All right. Thank you guys for coming.
Yeah. Thank you guys.
Thank you so much for going.
Thank you.
Yeah, of course.
Yeah, thank you.
No problem. Take care.
Have a good night.
You too. Bye.